LabToGo3 is an open source workstation for planar chromatography. It can be used for sample application, development of the samples, and visualization of the results. All parts can be controlled via a web application that you can see here. It is much cheaper than the technology commonly used in chromatography, which makes it perfect for student projects, schools and scholars, as well as individuals interested in testing food or water quality. But how does it work? Here you can see a stepper motor, which turns a trapezoidal screw, which in return moves the card, pressing a syringe down. If the syringe is pressed down, a pressure will generate in the system. To measure the pressure, we have here a force sensor. So if we press down on the syringe, we will measure a higher force than we don't press. And also we can quantify the pressure that way. Now that we have a pressure in our system, we can open a valve, resulting in a fluid ejection. To move the nozzle relative to the plate, we have another stepper motor that moves this plate holder in this direction, the Y direction, and also the nozzle in X direction. That way we can place our samples accordingly. But how do we control everything?
The brain of the machine relies on two very well-known electronic boards. The one on the right is the Arduino Mega, and on the left we have the Raspberry Pi version 4. The rest of the boards are underlying electronics that help, for example, to get the right amount of power from the lamp or to convert that power into light. The Arduino receives all the commands from the Raspberry and controls the actuation of the system. That is, coordinate the movements, take measures from different sensors, and turn on the LEDs, and so on. On the other side, the Raspberry hosts a web application called OCLab3 version 3 that has a friendly user interface that will allow anyone who wants to use the OCLab to set all the different configurations to get a proper sample application, development, or detection process.
For the future of the project, on one hand, we try to improve the machine where we can. We are working on making the fluid ejection more precise, thinking about applying different samples automatically, and optimizing all the machine parts and the code, as well as the user experience. On the other hand, we want to make a product that is affordable for everyone, so that we can create a community of users. Because of this open source philosophy, everybody can distribute. The competition with state-of-the-art machines could result in lower prices in general, spreading planar chromatography around the world and resulting in better food and water quality overall.
I will show you now very quickly how to set a sample application on OCLab3 version 3. The first time that we open the app, we will have to create an account and then connect with it. It is a very simple procedure. After that, we have access to the main window of the app. We see that we have a list of devices here. We select our OCLab from this list and we connect. There's a toolbar on the left of our screen. This is where we will find all the different tools that OCLab3 has. To do a sample application, we simply click on Sample Application inside of Application. We can now configure the sample application that we want to make. I will mainly use the initial configurations that OCLab3 already settled for me, but instead of 2 bands, I will apply 5 bands. I set the volume for each of them, then I save it and let's apply. After some minutes, the application will be completed.
To start the development process, you have to go onto the development page. You will find it on the left tab. Click development and you will see the development page. Here you have some settings for loading the syringe. You can just select how much volume is in the syringe, and then move the syringe push of the syringe pump accordingly. Also, you can rinse the system either automatic by selecting volume and speed of the syringe pump, or by manually opening the valve and then rinsing by pressing the syringe by hand and close the valve again. Then you have some options. Here you have the speed of the nozzle movement. It's in millimeter per second. You also have the pressure you can set. This is the pressure that will be produced before the valve is opened, so before a band is developed or one application is done. Then you have the temperature. If the temperature is set to 0, the heating foil will not be heated. If you set it to 70 or so, then the heating will begin before the development starts. Then you also have the option to select the nozzle type or diameter you have currently equipped. Furthermore, you can select the plate size of the plate you are using. We are always using 100 times 100, so this is the standard option. You have the option to set the location of the development. For example, if you type 50 here, you will see it here in the plate preview. If I specify it as 3, then it's up here at 3. This depends on the development chamber you are using. Furthermore, you have some options for the volume. You can specify the volume you want to apply. Let's say 1000, and I want 10 applications, so this would be 100 microliters per application. You also can specify the waiting time. The waiting time is the time the machine will wait after application of one application. You can also specify the fluid and add some descriptions or notes. Here in this line you can check or uncheck this option. This will result in, if it's checked, the ejection of fluid will be done on both ways, so from left to right and from right to left. If you only have it if you have it unchecked, then it will only do it in one way, so maybe from left to right. Okay, now we have that out of the way. Let's go to the flow rate settings. Here you can select how many points you want for this approximation, and you can specify the flow rate over the location X. If this is the X direction and this is the flow rate increase, let's say. So let's say we want to have more flow at the end, more volume at the end, because we saw that the band didn't look that great. We have the option to increase the flow at the end and make a proper development this way. Okay, now we have that. We can save the method by pressing new, selecting a name, save 5, and save it. Also we can delete saved methods, and we can also export those to a CSV file, an Excel sheet with all the options now inside. If you press start, stop, or pause, the machine will start the development, stop or pause the development. Furthermore, you have to make sure to put the development chamber on top of the plate, and also set the nozzle height accordingly. Oh, one thing is that the flow rate is calculated internally, and it will show here. This is a good way to make the development methods comparable, because the flow rate is in the end the main factor of the fluid stream that goes out of the nozzle and properties of it. So always check the flow rate, and if you see that the flow is not enough, you can change the volume or the speed, or the applications as well, to change the flow rate.